Hi guys, welcome to 13 Universal. This is A-level biology with Shedro. Okay, we are starting a new series today with on nucleic acids and protein synthesis, and this is series one. Welcome to this series. Check our videos and then all the series from cell structure to mitotic cell cycle. We have a lot of educative stuff, loaded videos that will help you scaffold navigate your growth and your development. Okay, it's very important, so make sure you check it and then get back to me. Okay, so let's look at some of these questions. Come with me, bring your writing pad. Okay, get your writing pad and your pen. Let's look at the first question here. Says which of the letters shows the correct order of the steps in the experiment. Okay, let's see. This is an experiment. Okay, just want you to try it. Try this question. I want to know how far you know this topic. It says DNA. You can just try it. Okay, pause and try. Look at what it says here. It says DNA is said to replicate in a semi-conservative way. Result of Messerschmitt and Stars. Messerschmitt and Stars, they worked on they just advanced the work of Watson and Crick. Okay, so their work, Messerschmitt and Stars experiment, gave overwhelming support to this theory. All right, the theory was from Watson and Crick. They used E. coli, which has a generation time of 15 minutes. Here are the steps in the experiment, but they are in the wrong order. So this is in the wrong order. So the question is, which sequence of letters shows the correct order of steps in the experiment? Okay, let's look at it. Okay, so of course, where this all starts is, of course, you know that in this experiment they used two isotopes of nitrogen. There are two isotopes of nitrogen. This is N15, which is heavy isotope. Okay, and then N14 is the light isotope. Okay, so the first thing in this experiment is that the bacteria was first of all grown. So you first of all grow the bacteria. So I think that should be S. Bacteria grown in a N15 medium for many generations. That's where it starts. That's where it starts. You grow it. Okay, so if you go to the option, the one that starts with S is most likely suitable answer. So A and B eliminated. All right, let's go to C and D. Okay, we're working on C and D. Come on, guys, watch this so you can see C and D. So let's go on. Now, once you have grown this bacteria in a medium, what happens is, because the new DNA material, remember that you are growing it in a medium that contains N15. Nitrogen is important, is a part of nitrogenous bases. So nitrogenous bases is the part of nucleotides. Nucleotides are building blocks for DNA. Okay, so that means that if you grow bacteria in a medium that has N15, after several generations, all right, the DNA strands would have been made with N15. Do you get? So because the nucleotides used to make them will be N15, so all your DNA will be N15. Okay, I think I'm slow a bit, but don't worry, trying to get you into this. All right, I'm not going to go deeper. Right, this is N15. So are you following? Hmm? Yeah, if you're following, let me know. All right, N15. So you see, so after so many years, but it will appear like this. So we expect that after so many, the next thing that happen is that P is that all bacterium will contain N15. Yeah. So if you come to C and D now, the next one is P. If somebody chooses R, bacteria contains either no, it's not yet time. So once you grow them in this, all right, for many generations, we expect that they will all have this, right? Okay, that's P. So from C now, from this, you will know that the answer is going to be C. But if you continue to verify whether it is correct, now after P, that's all bacteria contain 15 N15 isotope. So you go to the next one, which is T. T says all bacteria transfer. Yeah, this is when you transfer it to N15 N14 medium, and then sample every 50 minutes. Okay, yeah, this is logic is correct. Then after that, Q says all bacteria contain. So after the first generation time, the bacteria, this is why I waited here. Okay, so after, when you grow it in N15 medium, you know that this N14, all right, N15, these two N15 will separate and they will be used as template. Do you get? So now we are growing them in N14, so all of them will be used. Of course, the new DNA molecules will have N14, pardon me. Okay, N14, and this shows a hybrid DNA molecule. This also proves semi-consecutive replication. All right, because in semi-consecutive replication, don't forget that each new DNA molecule form must have a strand of old parent material and a newly synthesized DNA material. Okay, so that is why Q is correct. All bacteria contain hybrid after first generation, and R says the last one, bacteria contain either. Yeah, if you continue to divide. So this is C. Don't worry, we're gonna do a video on this, okay, and then post it too. So the answer is should be what? C. Let's go to the next question. Come on, guys, let's go. What is the minimum number of base substitutions required to change the nucleotide sequence of HB normal? Now, this HB normal, hemoglobin, the gene that codes for hemoglobin. Hemoglobin is a pigment that transports oxygen. So this gene is HBA. Now it is called HBB. All right. Allele to HBS. This is the abnormal allele. When the normal allele undergoes base substitution, it gives us abnormal. This codes for sickle cell allele. Okay. Now it's asking us what's the base substitution. Of course, guys, if you're conversant with this, you know that it's just a base substitution. All right. So the original base triplet is CTT. All right. This is CTT. So CTT undergoes base substitution. So you see, this thymine is replaced by adenine. Adenine. So when it's replaced by adenine A, that thing up here, this look at this. This is A. It's gonna give us, all right, after mutation, all right, we're gonna have only one base is changed. That's thymine, and it's replaced with adenine. So we're gonna have C80. Okay. So you notice that this CTT normally codes for glutamic acid, which is polar, right? So it's now being replaced by C80, which codes for valine, which is non-polar. Anyway, I'm not going to go further than that. We're going to see questions that will explain that. But see what I'm trying to say is one base substitution. Okay, one. Adenine replaces thymine. Okay, and instead of having CTT, we're having C80. So the answer is 1. Do you get? All right. Next question. In a DNA molecule, the base sequence AGT codes for the amino acid serine. Okay, let's see. What is the base sequence of the anticodon on tRNA to which serine becomes attached? Okay, guys. That is it there. But I want you to know something. All right, this is just a fast one. This is the base sequence AGT in a DNA. Don't forget that. I want to say this. The base sequence, all right, in a DNA is complementary to that of mRNA. That is, the base triplet in DNA is complementary to the codon on mRNA. The same thing, the anticodon on tRNA is also complementary. You get to the codon on mRNA. So this is DNA, right? Whatever that is on DNA. Sorry for it. Okay. Sorry. This is DNA. We have mRNA because DNA is used to make mRNA. during transcription. Remember, DNA is used as a template to make RNA during transcription. Okay, so this is tRNA. Okay, don't worry, we're going to get you videos on that. tRNA. Yeah, look at this. See this, guys. This whatever that is here is complementary. So if I have, for example, AGT, I just want to do this just to show you in case you see this question next time. All right, know what to do. This, you shouldn't spend more than 2 seconds. This is AGT. AGT. All right, that's AGT. Now, of course, it's complementary to mRNA. This is mRNA. Okay. This A, okay, base pairs with goes with U. Okay, G goes with C. T goes with A. So this is UCA. That's the codon on messenger RNA. Is UCA. And if you check in the option, you see there, but that's not correct. Now for transfer RNA, of course, messenger RNA base pairs with, of course, complementary with codon on transfer RNA. This is going to be same A, all right? I want to pay attention. And this is going to be C on messenger RNA goes with G, and that A goes with U. Okay, good. Look at this. Look at the trick here. DNA-based triplets AGT, mRNA codon is UCA. UCA is complementary to this, right? Now, tRNA-based anticodon is AG, which is complementary to this. So this is complementary to this. This DNA is complementary to this. tRNA is complementary to messenger RNA. Therefore, C, DNA and transfer RNA should be similar. So anytime they give you anything on DNA, remove T. That is, if it has T, once you remove T, replace it with U, that will give you the tRNA anticodon. Simple. So if at the beginning, once I see this, I just remove T and put U. Is that okay? That gives me AG. AG should be the answer. That's it. All right. But it's okay. I had to do that explanation to get into the, all right, the mental process of that, the logical, the thought process. And look at this. What would be the result of analyzing part of a DNA molecule? All right. So is A correct? DNA molecule is made up of nucleotides. A nucleotide consists of okay, let me draw this. Watch this. Okay, sorry. Yeah, just watch this. This is a nucleotide. I'm trying to draw. Yeah, sorry for the diagram. This is base. Okay, now watch this. So this is a nucleotide. It has a pentose sugar, a nitrogenous base, and a phosphate group. Okay, let's go. A says hexose sugar. It doesn't have hexose sugar. This cancels it. All right. Yeah, it doesn't have hexose 7 carbon sugar. But what we have in DNA is pentose 5 carbon sugar. All right. So B says nucleotides and phosphates are equal proportion. Nucleotides, okay. So if I have one nucleotide, I'll have one phosphate. That makes sense. It says the second one says an equal number of 8 adenine and cytosine. No, adenine and cytosine, they don't have equal number because they do not base pair. Adenine base pairs with thymine in DNA, while cytosine base pairs with guanine. So this is wrong. Okay, C says pentose sugar and phosphate equal proportion. Yeah, pentose sugar. This is pentose sugar. So in every nucleotide, I have one pentose sugar and I have one phosphate. That's correct. And then the next sentence says, and an equal number of adenine and thymine. Yeah, adenine base pairs with thymine. Okay, so if I have 10 adenine, I'm going to have 10 thymine. So this C looks correct. What do you think? I'm listening to you. What do you think? Okay, sorry for the background noise. Okay, now this says twice as many phosphates as pentose sugars and an equal number of adenine and guanine. Of course, adenine and guanine do not base pair. Okay, so C is the correct answer. Okay, Remember, we help you to build technical skills. We have to scaffold for you step by step on how to solve questions. Okay, so watch how we do it. 5 says the mechanism of action of 4 drugs that inhibit DNA replication is stated below. Yeah, see that, see that. If you are a pharmacist, all right, and you're building a drug to save lives, building a drug to destroy pathogen, okay, so you must know what your drug is targeted to do. Every drug or antibiotics has a target site, all right, in the pathogen. So look at what we have here. The mechanism of action of 4 drugs that inhibit DNA replication is stated below. All right. Okay. Just a minute. Yeah. So look at it. The mechanism of action of 4 drugs that inhibit DNA replication is stated below. Look at this. Call the name of this guy. What's the name of this drug? Alpha-dicholine, whatever his name is. Alpha-dicholine inhibits DNA polymerase. So polymerase catalyzes the entire process, okay, of replication. Helps to copy the template strand, addition of nucleotides to the growing chain, and of course forms phosphodiester bond. Okay, let's see. Cytarabine is converted into a molecule that substitutes for a DNA nucleotide. So this guy converts, this is converted to a molecule that substitutes, so it will not allow DNA nucleotide, and also it inhibits repair mechanism. Why this is involved in this? Okay, inhibits unwinding process, so it will be inhibiting helicase. Then hydroxycarbamide inhibits an enzyme involved in production of deoxyribonucleotide. Pause this and try it. So the question is, which role correctly matches a drug to an explanation of mechanism of action? Okay, let's go. Let's do it. Check this out. Decrease pool of available nucleotides inhibits chain elongation. So which one? Remember that. Look at this. Hydroxycarbamide is involved with the production of deoxyribonucleotides. So deoxyribonucleotides are the building block. They are the building block, right, for DNA. So if any enzyme that decreases it will affect elongation. Okay, so it's only D, because that's the enzyme that does that. So the rest is the answer, but you can confirm. You can check this DNA strands not available as template, so they do not unwind. So you can check epirubicin inhibits DNA unwinding. Check the rest. SATA. This talks about DNA damage. Okay, the one that prevents repair DNA repair mechanism. Satarabine. Look at it here. Okay. Okay. So pharmacists are very intelligent guys. So the answer should be D. Let's go to the next question. The following event occurs during transcription. Bonds break between complementary bases. Bonds form. This is form is a typo. Form between a complementary bases. Sugar phosphate bonds form free nucleotide. Okay. Now, before mRNA leaves the nucleus, which event will have occurred twice? What do you think? Chat me now. What do you think is the answer? Okay, you're correct. Let's do it. So remember, guys, remember that there's a bond break. This is hydrogen bond breaking between complementary bases. Complementary bases are bonded, right, using hydrogen. Okay, remember that during transcription, right, if this is DNA, right, DNA double helix, remember, will unwind. This is double helix. So why is wounded is because of hydrogen bond between bases. Okay, so unwind here so that we can have template. They separate. So I have template. All right. So they're separated. So we have of course, let's assume if you have A here, right? So there's going to be C because A is complementary to T, sorry. So they form two hydrogen bonds. Remember, if I have C here, so I'm going to have G here, and they pair by forming hydrogen bonds, right? So and remember that during this replication, okay, So the hydrogen bonds here break so that we can have this. But you see, when else does it break? When else does it break? The first time it breaks is when this unwinds a section of DNA. Not every part of DNA unwinds. Okay, let me show you something. Not every section. Let me make it bigger. Come with me. So if you have this is DNA, giant molecule, right? A section of DNA is just needed. That's just all we needed is just a section of DNA. So that section unwinds is only the section that unwinds, right? Let's assume that this is the section that unwinds. It's only the section that unwinds. The rest do not unwind. The rest parts, okay, stay unwound. Okay. So if you have, of course, look at this. This is let's assume this is template strand and you have A here. Okay. Yes, adenine. You have C here. You have you have G here, whatever. Let's assume this is it, and so forth. So when the RNA nucleotide pair with that, okay, this is A. The nucleotide with T will pair with A. All right. This is no. We don't have we do not have nucleotides with T. Okay, is either what we have is nucleotide with U. So this is going to be A pairs with U. Okay, and the other one is C. We're going to have a nucleotide here, which is G pairs with C. Sorry, this is G. Okay. All right, and this is G. G will pair with another nucleotide that have C. Is that okay? So they form hydrogen bond. Remember that the hydrogen bonds are formed between these bases. For example, here 3 hydrogen bonds form between cytosine and guanine, and guanine, and then between here and this is 2 hydrogen bond. Please don't forget that this is guanine, okay, and this is cytosine. So now, of course, RNA polymerase will link them up to form a strand of messenger RNA. Okay. All right. Now look at it. Okay. After this, messenger RNA will leave. So bonds are broken first of all when this DNA unwinds. Okay. Bonds between the two strands are broken. But after the unwinding process, after messenger RNA is synthesized, hydrogen bond between the bases on messenger RNA and the non-template strand must be broken. So bonds are broken twice. Okay. Bonds formed between complementary bases. Bonds are formed twice. The first one is bonds formed between the bases on messenger RNA. This basis, the RNA nucleotides that forms messenger RNA, between them and their complementary bases on the template strand. That's the first time. The second time it forms, bonds are formed is when this winds up back. Of course, after transcription, DNA will wind up back. Okay, and of course, when it winds up back, bonds between this strand, hydrogen based on this strand, and that strand will form. Okay, so bonds are formed twice. Bonds are broken twice. Sugar phosphate backbones forms once. When RNA polymerase joins the nucleotide to form mRNA. Okay. Now, free nucleotides pair up with complementary bases just once. That's when they pair up. So the ones that occur twice is bonds breaking and bonds formation. So bonds, the first time is when this unwinds. That's the first time it breaks. The second time it breaks is when this messenger RNA leaves this nucleus. The bond between the bases on the messenger RNA and the template strand must be broken, right? So the first time it is formed is when the nucleotide align along the template strand. They form hydrogen bond with their complementary bases. That's the first time bond is formed. The second time bonds are formed is when this DNA winds up back. Okay, so bonds are formed between the basis on the 2 template strand. So therefore the answer is what? A, 1 and 2. All right, let's go to the next question, guys. Which type of sugar and types of bonds are found in DNA molecule? Yeah, of course, pentose sugar. Pentose sugars are not reducing sugar. They are not non-reducing sugar. They're just reducing sugar. So C and D are selected. Now there are 2 bonds that are formed. Covalent bond, phosphodiester bond formed between nucleotides, and hydrogen bonds formed between complementary bases. Peptide bond is not formed in DNA. It's found in protein between 2 amino acids. Right. So the answer is C. Okay. The graphs represent the changes in the quantity. Okay, this question is on mitotic cell cycle. We have done it before, all right? On one of the series in mitosis. Okay, please watch it. But you see, just to mention here, X is when the DNA content decreases. Okay, that is when the cell is splitting. Is in telophase. Okay, because cytokinesis starts in telophase. But just watch the video there, right? Now the table shows tRNA anticodon for 4 amino acids. Look at it. All right. So the question says, what was the sequence of bases on DNA from which this was from? All right. You can pause and look at it. So these are the amino acids. These are the anticodon. All right. And this is the polypeptide chain. Okay. Cell makes a polypeptide. This is the first amino acid. This is the sequence of the amino acids. The first one is glutamic acid. Second one is asparagine. Third one is threonine. The proline. Okay, what was the sequence of bases on DNA from which this was from? Remember, I told you once you know the sequence of bases, the anticodon of tRNA is similar to that of DNA. Just whatever you see, you put T. If you're moving from tRNA to DNA, if you're moving from DNA to tRNA, do the same thing. Whatever you see T, put U. So glutamic acid, what's the code? CUU. For that of DNA to be CTT. Okay, I'll come to option, the one that has CTT. Okay, this option C and D. So A and B eliminated, CTT. Okay, let's go to the next amino acid, asparagine. Asparagine is UUA. That will be TTA. Which one has TTA? Both of them have TTA. Okay, TTA. TTA. Okay, let's go to the next one, threonine. Threonine is UGG, which is TGG. So this is TGG. This is TCC. So this is wrong, right? So this is TGG. So C is answer. All right. What does the enzyme RNA polymerase synthesize? RNA polymerase, guys, is the enzyme that catalyzes transcription. You know, I said it now. Have you forgotten? It just links up RNA nucleotides, linking them up, forming phosphodiester bond between them. So this guy, the question says, A says a polypeptide from, no, this it doesn't catalyze translation. Okay, this is translation, synthesis of polypeptide using RNA as template. That's translation. A strand of DNA from mRNA. Okay, strand of DNA from, this is called reverse transcription. So the enzyme that catalyzes this using RNA, mRNA to form DNA is reverse transcriptase. mRNA from a DNA. Yeah, that's RNA. This sounds like it. So it uses DNA as a template to make RNA. That's RNA polymerase. That's transcription. Okay. This is mRNA from it. No. So the answer is this: using DNA as a template to make RNA. That is transcription. Okay. So the answer is C. All right. Thank you so much, and please don't forget that if you have questions, post it. Like you to like the video, subscribe, follow us. So the next one will be on, that series 2. See you. Bye. Don't forget this is schedule. Bye bye.